Hello, welcome back to the third part of the replication series. In the first part of the replication, we have discussed about the enzymes that are involved in the process of replication. Second part was really regarding the replication in prokaryote, and this third part is regarding the replication in eukaryote. The general process of replication in both the prokaryotes as well as in the eukaryotes is same, but the only difference is the factors. Like the enzymes are same, but what are the factors, the name of the factors are different. We will be discussing the main thing in this part, that is what is the difference in this prokaryote eukaryote, and what are the enzymes that are involved in, what are the protein that are involved in E. coli, and what that enzyme is called in case of the eukaryotes. So we will be discussing all this in detail, and let's begin. Before that, I want to quickly recap the what are the functions of the enzyme. So let's begin with that. First of all, the first thing is that mainly what will happen, the two strands of the DNA need to be opened so that the process of replication can take place. For that, the enzyme which is involved is the helicase enzyme. So the helicase enzyme will bind to the one part of the DNA strand, and it will start opening the strand and form a replication fork. This is known as the replication fork, and the DNA will start opening. Two single strand DNA will be formed. As the DNA is now in the single stranded state, and there is high chances of them to be degraded by the nuclease enzyme, because nuclease enzymes are also present, so they can degrade it. For that, another protein which is known as single strand binding protein binds to the single strands. You can see here, they will bind to the single strand DNA and provide stability to them. This is the second enzyme which is second protein which is involved. First is known as the helicase. Next, moving on, as the replication fork keeps on moving, there will be positive supercoiling towards the, as the replication fork move down. As a result, it will create tension on the DNA strand. To release those tension, or to make the replication process go smoothly, that's why another enzyme which is involved is known as the topoisomerase enzyme, which is the third enzyme to be involved. Okay, they will release the supercoiling so that the replication fork can move easily. Next, what happened after all this thing, the primer will be attached to the one end, and you know that the primer provide 3' OH end, free 3' OH end, to which the DNA polymerase will bind to it. Okay, the DNA polymerase will bind to the primer, and the synthesis of the new strand takes place. And the DNA polymerase synthesis form two strands. One is known as the leading strand, which runs from, the parental strand runs from 3' to 5'. So the leading strand will be formed, that is the complementary strand will be forming in the 5' to 3' direction. The leading strand, parental strand is 3' to 5', you can see, and the complementary strand which will be formed is from the 5' to 3', as the DNA polymerase will be binding here, so it will move from the 5' to 3' direction. And one more feature of the leading strand is that it will form in a continuous manner. As you can see here, it will form in the continuous manner of the replication. Okay. Next, coming to another type of strand which is formed is known as the lagging strand. As you all know that the DNA polymerase will always move in the 5' to 3' direction. Okay. So 5' to 3' direction it will move, and as it will be binding here, so here it is opposite, the strand is opposite here, it is 5' to 3', it is opposite, and the newly formed strand will be from 3' to 5'. So it will be in the opposite direction to that of the DNA polymerase. That's why the breakage in the strand will take place. Breakage means Okazaki fragment will be formed. That is, multiple primer will be required for forming of each strand. That's why the lagging strand is formed. That is, it is a discontinuous strand. Why it is discontinuous? Because it is DNA polymerase is acted on 5' to 3' direction, but these strands move from the opposite direction. Okay, it moves in the opposite direction. That's why the strands is in the form of a discontinuous manner. Okay. Next, finally, what happens when they will come to the termination sequence, then the process of replication will be completed, and then all the primer, everything, all the enzymes will be removed, and DNA ligase along with that of the DNA polymerase 1 in case of prokaryote, they will join the seal, and then a complete replication or the complete new strands of the DNA will be formed. As you all know that the DNA is a semi-conservative, that is, one strand will be of parental strand, another strand will be of new strand. Hope with this you are clear with the enzymes and what are the role. Now we will be dealing with the difference, that what are the DNA, what are the protein that are involved in the E. coli, and what are the protein involved in eukaryotic protein, and what is its function. The proteins are only, the names are different, but the functions are same, and by this you will be getting a revision of E. coli replication. First comes to the DNA-A. If you remember, the DNA-A protein in E. coli was responsible for recognition of the origin of replication. As you know, the replication process start from a ori side. For that, a protein is needed to recognize that this is the origin site, and here the primer need to be binded and the replication process need to be continued. For that, a protein is required. In E. coli it is DNA-A, and in case of eukaryotic protein it is ORC protein. Okay. Next, coming to the topoisomerase. In case of eukaryotic protein, topoisomerase 1 and 2 are involved in releasing the supercoils which are formed. But in E. coli, the gyrase enzyme is involved. Gyrase protein is involved. Okay. Next, coming to the helicase. Helicase will bind so that the opening of the strand will take place. In case of E. coli, DNA-B will do that function. You will be in the last video I have mentioned the what are the role of the each protein in E. coli. Next, in case of eukaryotic, this work is done by MCF, that is, it acts as a helicase enzyme and it will start opening the strand for the process of replication. And you know, once the strands are open, there need to be stabilization. For stabilization of the strand, there need to be a certain protein so that it will, the strands does not get degraded. For that, in case of E. coli, SSB proteins are involved. In case of eukaryote, RPA proteins are involved. Okay, they maintain the single strand. Next, coming to the DNA polymerase binds to that of the strands of the DNA by the help of a clamp. Okay, by the help of a clamp or by the help of certain subunit. Okay, so that they can, so that the DNA gets fixed, hold properly, and the primers get attached properly. Otherwise, the DNA will move away, slide away, as it is a fluid state. So as the DNA will move away, that's why to hold the DNA to proper, so that the primer can be added, that's why certain subunits are present in case of DNA polymerase, like that of the gamma complex in case of E. coli, and RFC in case of eukaryotic protein, which is responsible for loading the clamp. What is the role of the clamp? Is to hold the DNA polymerase onto the DNA strand, so that the primers can be added and the replication process goes on smoothly. Next, moving to the next one, that is synthesis of the leading. Now the DNA polymerase is loaded on the strand. Now what will happen? Synthesis of leading strand and lagging strand will take place. In case of E. coli, polymerase 3, only one polymerase is responsible for the formation of both the leading as well as the lagging strand. Whereas in case of eukaryotes, that is, polymerase epsilon is responsible for formation of leading strand, and polymerase delta is responsible for formation of lagging strand. Okay. Next, coming to the clamp. Already I know the function of clamp. In case of E. coli, beta clamp is present, and in case of eukaryote, pCNA clamp is present, which holds the DNA polymerase. Already the function is known. Next, coming to the primase. What is the role of the primase? Is to form the primer, is to synthesize primer, so that the DNA polymerase will bind and the process continues. Okay. Next, at the last, after the synthesis of all the leading and lagging strand, finally the sealing of the strand should take place, which is performed by DNA ligase that is in both prokaryote as well as in eukaryote, DNA ligase is responsible for joining of the Okazaki fragment or the discontinuous fragment. Next, after that, the removal of the primer is necessary in case of eukaryote, which is done by FEN1. FEN1 is that it removes the primer, okay, and in case of polymerase 1, it is in E. coli, it is responsible for sealing the gap. So this is a very, very important table, and this is only asked in exam, that is which protein in E. coli, which and the functions will be given. You need to match the following, certain sort of that type of questions will be given to confuse that. So be prepared for that type of question. If you go thoroughly with this, very easy it is. If you go thoroughly, you will remember everything. First of all, first part of the video you go through that what enzyme is responsible for what activity, like helicase is doing what, topoisomerase is doing what. Then you need to distinguish what type of proteins are involved in this E. coli and what are in the eukaryote, so that it will be very easy for you. Next, moving to the difference between the replication of prokaryote and eukaryote. The main difference is that the prokaryote requires a single replication origin site, whereas eukaryote is very complex in structure, so multiple replication origin site will be required. This is very important. Okay. Next, coming to the rate, that is, as the single replication site is present, so naturally the process of replication will be more faster as compared to eukaryote, because many replication origin site will be there, so the process will be slower. And the DNA polymerase that are involved in prokaryotes are 5 type, 1, 2, 3, 4, 5, and in case of eukaryote there are 14, like delta, alpha, delta, epsilon, gamma, and so on. And primer remover is done by DNA polymerase 1 in case of prokaryote, and in case of eukaryotes RNase H, or you can say FEN1 is doing that function. Then one interesting fact is that telomerase. What is telomerase? We will be dealing in the next part of the video when we will be reading about the telomerase replication, because telomerase is a part of eukaryotic chromosome. Okay. We will be dealing with that in the next part in detail. For now, you understand that in case of prokaryote, the telomerase is absent, whereas in eukaryotes, the telomerase is present. And the length of the primer in case of prokaryote is 50, whereas in case of eukaryote, it is 9. So this is all about this. Two tables are very, very important regarding exam point of view. And for better understanding, do watch the first part of the video, second, and then come to this part of the video, so that all the concept regarding replication will be clear. Next, I already have told you that the eukaryotes are mainly linear, have linear DNA, whereas the prokaryotes have the circular DNA. And in case of eukaryote, multiple origin site will be there. So for each origin site, each replication fork will be present, and there will be a bulging in the where the replication takes place, and ORIC will be, first ORIC will come, it will recognize the origin site. Okay. Then what will happen? The MCM will come, that is the helicase, it will start opening the strand. Okay. Once it start opening the strand, the RPA protein, it will bind to the single strand to provide stability to it. Then the topoisomerase 1 and 2 is responsible for what? For clearing the supercoiling. Okay. Next, RFC is responsible for loading the clamp, so that the DNA polymerase holds tightly. And next, polymerase alpha will load the primers, and then the starting replication will start, and finally after the replication process, the FEN1 will remove the primer. And these steps are done by 3 steps, that is initiation, elongation, termination. I have already mentioned it again and again, that what are the process, these are all written again. And one thing you to be noted down in the termination process is that although the termination is completed after this removal of the primer and all thing, but still there is a certain part in the DNA that is not been synthesized, that is the telomere part, that is known as the extra part of the eukaryotic chromosome. That is mainly done by the telomerase enzyme. So what is that? We will be discussing in the next part of the video. So this is the overall picture of what the replication takes place, how the DNA, how the enzymes and all are responsible for the replication process. The process of replication, again I am telling, the process of replication is same for both the prokaryote, eukaryote. Only there is a difference in the enzyme and the protein that are involved, and extra one thing that is present in the eukaryote is the telomere. And what is telomere, as well as what is telomere replication, and what are the inhibitors of the replication, which is very important again for the exam point of view, we will be discussing in the next part of the video. Hope this video is very fruitful for you, and once again I am trying to tell you that do watch the first two part and then this part for the overall clear picture of replication, and do share among your friends as much as you can, because this replication, transcription, translation are very basic and people think that they are very difficult and they skip this part, but molecular biology, this part is only the key fundamental thing. If you are good in this part, then nothing can, no exam or no question you can be unsolved in this. Okay, you can solve any type of question regarding this, and be clear with the fundamental, that is what enzyme is doing what thing, and everything will be good. So if you have if you like this video, do like, okay, hit the bell icon so that you get notified whatever video I am uploading, and share among your friends and subscribe if you are new to my channel. Thank you and have a nice day.